So the Kelvin cycle proceeds in like three stages. The first stage is that is carboxylation, during which CO2 combines with the RUBP, that is ribulose 1,5 bisphosphate. Okay. Then second stage is reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH. Okay. Then regeneration, during which third step regeneration, during which the CO2 acceptor ribulose 1,5 bisphosphate is formed again, so that the cycle continues. So the carboxylation is the most crucial step of the Kelvin cycle, where CO2 is utilized for the carboxylation of RUBP. Now, two molecules of 3-PJ is formed. Second is reduction, where reduction step involve utilization of two molecules of ATP for phosphorylation and two of NADPH for reduction per CO2 molecule fixed. Now regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue interrupted. This involves 1 ATP. Now in and out, 6 CO2 is used for 1 glucose, 80 ATP is used for, and 12 NADPH is used to 12 NADP. Okay, now C4 pathway. C4 plants are special. C4 plants are special. They have the Kranz anatomy, and they tolerate higher temperature, response to high light intensity, lack process called photorespiration, have greater productivity of biomass. Now they have the the vascular bundles of the C4 plants are called the bundle sheath cells, and the leaves which have anatomy, these are Kranz anatomy. Now bundle sheath cells may form several layers around the vascular bundles, so they are characterized by having a large number of chloroplast thick walls, impervious to gaseous exchange, and no intercellular spaces. Okay, so C4 plants are maize and sorghum. Okay, now C4 plant is hatch and slick pathway. It is cyclic process. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate PEP is present in the mesophyll cells, and the enzyme responsible for the fixation is PEP carboxylase or pepcase. Okay. First is the mesophyll cell, second is the bundle sheath cells. Okay, the bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase rubisco but lack pepcase. Thus, the basic pathway that results in the formation of sugars, the Kelvin cycle, is common to the C3 and C4 plants. Now, photorespiration. So, in C3 plants, some O2 does bind to rubisco, and hence CO2 fixation is decreased. Here, the RUBP, instead of being converted to two molecules of PGA, binds with O2 to form one molecule of phosphoglycerate and phosphoglycolate, that is 2 carbon, in a pathway called photorespiration. Now in the photorespiratory pathway there is neither synthesis of sugars nor of ATP, rather it results in the release of O2 with the utilization of ATP. In C4 plants photorespiration does not occur, right? This is because they have mechanism that increases the concentration of CO2 at the enzyme site. Productivity and yields are better. Okay. The factors affecting photosynthesis given by Blackman's, that is law of limiting factors. As a plant photosynthesis, all these factors will simultaneously affect its rate. The first one is light. What is interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Now, carbon dioxide fixation. For C3 plant, it is 450 ml, okay, and for C4 plants it is less, that is 360 ml. Now, so this is why the current availability of CO2 level is limiting to the C3 plants. Now if it so 0.05% can cause an increase in CO2 fixation rates. Okay, and tomatoes and bell pepper, these are C3 plants. Okay, now temperature. The temperature optimum for photosynthesis of different plants also depend. Okay, tropical plants have a higher temperature optimum than the plants adapted to temperate climates. Higher temperature optimum, right? Now C4 plants respond to higher temperatures and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. Now water.